The REACT project aims to develop a sterile insect technique application to combat Bactrocera dorsalis and Bactrocera zonata in Europe. The French overseas territory of Réunion Island in the Indian Ocean plays a key role in the project. Both fruit flies have invaded the island, making it a natural laboratory to study invasion dynamics.
I'm Hélène Delotte. I'm a researcher at CIRAD in La Réunion Islands. We are the first country in Europe where we had Bactrocera dorsalis. We had an invasion in 2017, and since then it's really stabilized population that ripped out Bactrocera zonata that was also an invasive species present here. So it was very important for us to study more and to try to find solutions to be able to export fruits, for example. We needed to study why are they so invasive in the world. A long time ago, there were only one Tephrididae known on fruits on Réunion Island. It was Ceratitis catuari, which is an endemic fruit flies of the Mascarene and present here at very low numbers. But then afterwards, successive invasion came onto the island, unfortunately, on the fruits. Ceratitis kilisi, then Ceratitis capitata, the mayfly, and most recently in the year 2000, Bactrocera zonata. And in 2017, we had dorsalis. Unfortunately, Bactrocera zonata for itself and dorsalis have the exactly same climatic niche. They like lowlands area of the islands where it's warmer, and they are also found on the same fruits. When Bactrocera dorsalis came, it really moved out Bactrocera zonata, and we think it's now the population of zonata that was very much present on the island is almost extinct. What is interesting for REACT to work on Réunion Island is because Bactrocera dorsalis just came in. So we are looking at an invasion process which could look like, mimic what would happen in Europe.
So, Europe can learn a lot in Réunion Island.
I would like to know if Bactrocera dorsalis is present all year round in the suburban areas, on the contrary of orchards where it's more monocropping. Like here you have the mango season right now, but half or three-quarters of the year you don't have any fruits. So dorsalis will or will not stay or disperse in this kind of area. This is one of our questions. Whereas in the suburban, we think that there are so many different trees that are fruiting at the same season, so we could maybe breed a population that we say the whole year round around at least. So be a nice spot for a propagate invasion and start of invasion and building up your populations.
The researchers at CIRAD are investigating how the invasive fruit fly spreads across the island. Where does it nest? How does it hibernate? Which host fruits does it like? And how does it compete with other fly species? It is not just fruit and vegetable plantations they look at, because pest invasions do not start there, but where they are introduced by humans, mainly in urban and suburban areas.
We are also looking at trapping at different altitudes, altitudinal ranges with Bactrocera dorsalis, and do the genomic study together with the Royal Museum of Tervuren in Belgium. So they are looking at the different population, if there are already adaptation between lowland and higher lands, or if this different population can be found already after a few years after the invasion, if there adaptation. This signal is very important for us to know if the flies have evolved or not, or adapt so quickly. So these traps are catching mostly males of Bactrocera dorsalis because it's a lure, methylogenol lure for the male catching. So they go into the trap and they'll be catching it. Here it's been hung for a week, so Amélie will open it and you will show all the dorsalis in it. Because we're in an orchard with lots of fruits, so we have many flies all the time now, summer. Unfortunately, Bactrocera dorsalis is a superfly because obviously only one small population were able to disperse all over the island at different altitude and adapt without any evolution since its first incursion in 2017. So one chance made them able to do it. So that's why it's very astonishing. Usually you need more propagate, more invasion. The more you have as in invading phases, the best you will adapt in one place usually. So it means that dorsalis for us in Europe If dorsalis, a population dorsalis is coming into a country and stay there, only a few individuals can colonize and stay there and develop. So we have to be more careful for sure and to be able to eradicate very quickly the first individual that will come as soon as we see them.
My name is Tanisha Phipps-Lave. I work in the DeLatte lab on the REACT project. I have worked on the competition experiment, so axionic versus symbiotic between dorsalis and capitata. And I also set up and collect the traps that we set. Twice a month we come and refill the torula, which is necessary. As you can see, there's so many other flies. We test for host performance for European endemic wild fruits for both sets of our fruit flies, Bactrocera dorsalis and Bactrocera zonata. And we do this in lab conditions. So the first thing that we would do is we set up two cages with 10 females that have already mated and are mature. And then we place the fruit inside for 25 hours. And then we come and recuperate or collect the fruit like this. Sorry, I don't want the flies to come out. And so we would take this fruit back to the lab and count the sites of oviposition. And after counting the sites, we would open each site and use a scale to estimate the number of eggs that were in each site. One single larva and place it within the slit that I just cut. So after that, we place the fruit in the sand. And then we place it in the 25 degree.
The various experiments with the flies in laboratory conditions follow strict protocols. The aim is to understand the conditions that encourage the spread of the pest so that fruit fly control can be made as effective as possible using sterilized insects. The experiments cover all stages of fly development, from egg to adult fly.
We started to check if there is a pupae. Yes, there is one. 0. Okay. We take the pupae one by one and weigh it. And then we take a tube with paper and we added nipagine benzoate to prevent fungus development. And we put inside the pupae and we wait then for the check for the emergence each 2 days.
I'm Laura Moquet. I study particularly 2 points. It's flight behavior, flight performance of insects, and also the mating behavior. We dissect insects and we observe the size of ovary of the females. We analyze the quantity of sperm that a male transfers to the female during the mating. We dissect these females. So I open the abdomen with my forceps. And I pull the ovipositor, and all our female organs come with, like this.
This is ovipositor. And here there are the two spermatheca in black. And if you follow the duct here, there are the fertilization chamber. All this part is the organ of stockage of the sperm of the females. So we can count and observe and count each spermatozoa of the inside the female spermatheca. It's important to know the quantity of sperm released by sterile males. Because the male is sterile, it can continue to produce sperm. Maybe it can transfer sperm during the first mating with female, and after that, there are no more sperm to transfer. If the females mate with sterile males, but there are no spermatozoa, maybe it can remate more rapidly after the first mating. So you need to take in consideration when we estimate the number of males to release during SIT.
In my field of work, I'm really very often looking at island biology. It is very super cool to work in an island because invasion came into the island and we can explore how the invasion, what it is doing, because we are in a closed environment. If there is only one single introduction lab for Bactrocera dorsalis, we can see how the population evolves, how it invades, and how it's possible to invade. So without a continental scale where we can have other invasion, we can look very much into details and follow the invasion very well.